Good morning. The bibliographic references and the publisher's source article are accessible in the description under the video. If you want to support the dissemination of scientific knowledge, I invite you to subscribe to this YouTube channel. It's free. Also, please, can you click on the thumb pointing towards up? Abstract: the use of polyphenols as adjuvants in lowering risk factors for various debilitating diseases has been investigated in recent years due to their possible antioxidant action. Polyphenols represent a fascinating and relatively new subject of research in nutraceuticals and nutrition, with interest rapidly expanding since they can help maintain health by controlling metabolism, weight, chronic diseases, and cell proliferation. Resveratrol is a phenolic compound found mostly in the pulp, peels, seeds, and stems of red grapes. It has a wide variety of biological actions and can be used to prevent the beginning of various diseases or manage their symptoms. Resveratrol can influence multiple inflammatory and non-inflammatory responses, protecting organs and tissues, thanks to its interaction with immune cells and its activity on silent inflammation regulator 1. This compound has anti-inflammatory, antioxidant, antipoptotic, neuroprotective, cardioprotective, anti-cancer and antiviral properties, making it a potential adjunct to traditional pharmaceutical therapy in public health. This review aims to provide a comprehensive analysis of resveratrol in terms of active biological effects and mechanism of action in modifying the immune cellular response to promote human psychophysical health. 1. Introduction. Diet and proper lifestyle play a key role in maintaining well-being and preventing diseases. In recent years, due to their potential antioxidant activity, the use of polyphenols as adjuvants in mitigating risk factors for various disabling diseases has been evaluated. Polyphenols, organic compounds abundant in plants, microalgae, fungi, and yeasts, represent an interesting and relatively new field of study for nutraceuticals and nutrition. Several studies show that the consumption of polyphenols can contribute to the maintenance of health by regulating metabolism, weight, chronic diseases, and cell proliferation. Experimental studies, clinical trials, and epidemiological research highlighted that polyphenols exert antioxidant and anti-inflammatory activities that could have beneficial effects on cardiovascular diseases, neurodegenerative disorders, cancer, and obesity. According to the World Health Organization, 52% of premature deaths in 2012 were attributable to non-communicable diseases, with more than 75% related to cardiovascular diseases, cancer, diabetes, and chronic respiratory diseases, which represent one of the main public health concerns. A modification in training, lifestyle, and diet may help to reduce the onset of these pathologies. The European Food Safety Authority has stated that the consumption of 5 mg/kg/day of polyphenols could help in the prevention of such diseases. Resveratrol, a phenolic compound found in red fruits and berries, in particular in grape pulp, skin, seeds, and stems, exhibits a wide range of highly active biological effects to counteract the onset of many diseases or to manage their symptoms. One of the possible mechanisms by which resveratrol plays its role in maintaining health is the suppression of inflammatory reactions by acting on immune cells. Resveratrol has antioxidant, anticarcinogenic, anti-inflammatory, neuroprotective, cardioprotective, and anti-aging properties. This natural flavonoid can mitigate toxicities related to chemoradiation therapy in normal tissues and enhance migration of cells toward injured areas, which is important from therapeutic standpoints. It also has modulatory effects on the tumor microenvironment. Furthermore, this polyphenol is also involved in the maintenance of liver metabolism, interacts and strengthens the immune system, and acts on the nervous system. Some studies report the neuroprotective effect of resveratrol against neurodegeneration, which sees its genesis in an impaired mitochondrial function. Resveratrol crosses the blood-brain barrier to counteract oxidative damage in hippocampal neurons and glial cells. It has been described that resveratrol can activate the silent inflammation regulator 1, a type of histone deacetylase, to protect neurons against apoptosis, inflammation, and oxidative stress in the treatment of neurological disorders such as Parkinson's disease. It also modulates cholinesterase activity and preserves dopaminergic neurons, which enhance learning and memory. It has been shown that resveratrol can reverse social dysfunctions. Recent studies have suggested the effect of resveratrol in improving panic and anxiety behaviors. Recent studies have shown that still beans, in particular trans-resveratrol and its glucoside, are able to bring numerous benefits to human health, showing antioxidant, cardioprotective, and antitumoral effects. Several studies prove the use of this phenolic compound in the treatment and prevention of diseases with tumor etiology, helping to stop uncontrolled cell growth and consequently suppress cancerous neoformations. The cardioprotective activity of resveratrol is associated with the inhibition of platelet aggregation and oxidation of low-density lipoproteins, followed by increased artery vascular relaxation. Several clinical studies have shown that resveratrol may be useful for diabetic patients. The therapeutic effect of this compound on diabetes is complex and includes several beneficial functions. The use of resveratrol, alone or in combination with modern antidiabetic therapies, may become an effective treatment for diabetes mellitus or its complications. Since resveratrol is a natural antibacterial agent, researchers are increasingly using it to treat acute inflammation and chronic illnesses. The biological properties of resveratrol tend to be closely related to a hormetic effect. Low levels are associated with positive effects, while excessive amounts may be toxic. Its hormetic qualities may be ascribed to its dose-dependent biphasic impact on cellular redox state, which is an antioxidant at low doses and a prooxidant at high concentrations. Despite a considerable number of human and animal studies demonstrating the therapeutic and protective qualities of resveratrol, there are not enough clinical trials revealing the contentious and deleterious effects of this flavonoid. The purpose of this review is to provide a detailed assessment of the active biological effects and of the mechanism of action of resveratrol in modulating the immune cellular response to improve human psychophysical health. 2. Resveratrol and immune cells. Resveratrol can enter the cell via passive diffusion, mediated endocytosis, or via transporter proteins binding to specific receptors, such as integrin receptor alpha-beta 3. It contributes to modulating innate and adaptive immunity, stimulating the activation of macrophages, T cells, and natural killer cells, and cooperating in the inhibitory regulation of CD4 plus CD25 plus T cells. This phenolic molecule can eliminate reactive oxygen species, inhibit the oxygen cycle, and activate many anti-inflammatory pathways, including silent information regulator 1. Silent information regulator 1 disrupts the toll-like receptor -4/nuclear factor kappa B/signal transducer and activator of transcription signal, decreasing the production of cytokines by immune cells and of pro-inflammatory mediators derived from macrophages or mast cells, such as platelet activation factor, tumor necrosis factor alpha, and histamine. Silent information regulator 1 is also involved in several molecular events, including metabolism, cancer, embryonic development, and immune tolerance. Ablation of silent information regulator 1 leads to increased activation of T cells and the onset of spontaneous autoimmune diseases. Due to its ability to activate silent information regulator 1, resveratrol is able to relieve inflammatory symptoms in several experimental models of autoimmune diseases, such as type I diabetes, encephalomyelitis, and rheumatoid arthritis. Activation of silent information regulator 1 by resveratrol results in inhibition of P65/REL acetylation, an NF-kappa B member, which is the main regulator of leukocyte activation and the signaling of inflammatory cytokines. This mechanism reduces the NF-kappa B-induced expression of inflammation factors, such as TNF-alpha, interleukin-1, IL-6, metalloproteinases -1 and -3, and cyclooxygenase 2. Resveratrol crosses the membrane by 3 mechanisms, namely passive diffusion, endocytosis via lipid rafts, or by carrier-mediated transport, binding to receptors such as integrin alpha-beta 3. Once in the cell, it activates silent information regulator 1, which inhibits REL acetylation and promotes inhibitor protein B degradation, lowering NF-κB-induced production of TNF-alpha, IL-1, IL-6, MMP, and cyclooxygenase 2. Protein kinase is activated by cyclic adenosine monophosphate levels, which activate silent information regulator 1. Silent information regulator 1 activity is controlled by AMP-activated protein kinase, which regulates cellular levels of nicotinamide adenine dinucleotide. Increased Na2+ levels activate silent inflammation regulator 1, which promotes decetylation and activation of peroxisome proliferator activated receptor gamma coactivator 1 alpha, which is downstream of AMP. The development of tumors and inflammation are linked. As a result, anti-inflammatory drugs can help to prevent cancer from growing. Resveratrol has been demonstrated to have cytotoxic and anti-tumor effects in vitro against a variety of cancers, including breast, ovarian, stomach, liver, thyroid, and prostate cancers. Resveratrol can act on the signal transduction pathways involved in angiogenesis, metastases, apoptosis, inflammation, and cell proliferation, affecting cancer development and progression. Resveratrol was used to counteract colorectal cancer in a mouse model in a 2020 study. Treatment increased in anti-inflammatory cells CD4+ deregulatory and CD4+ IL-10+, induced a decrease in pro-inflammatory cells T helper 1 and Th17, leading to a deceleration of colorectal cancer growth. The study of immunomodulation of the tumor microenvironment is becoming increasingly essential in the treatment of cancer patients. Several non-tumor cells, mostly endothelial cells, carcinogenic fibroblasts, and immune cells, primarily related to tumor macrophages, cytotoxic T cells, natural killer cells, B cells, Treg cells, and dendritic cells, make up the tumor microenvironment. These immune cells play a key role in the pathophysiology of cancer growth and progression. Resveratrol has recently been postulated as an immunomodulatory drug capable of stimulating immune cells in the tumor microenvironment or sensitizing tumor cells to immune cell cytotoxicity. Resveratrol has been proven to increase the production of anti-tumor cytokines, including interferon gamma and TNF-alpha, as well as to decrease the production of tumor growth factor beta. It can also encourage CD4+ T cells and macrophages to polarize towards anti-cancer cells, while reducing the invasion and polarization of immunosuppressive cells. 2. 1. Macrophages. Macrophages, immune cells that differentiate from monocytes, cooperate in the balance of innate and adaptive immunity. These cells form a heterogeneous pool of cells with a broad spectrum of biological activities depending on where they reside and the external inputs they receive. These immune cells have a wide range of pattern recognition receptors, allowing them to identify specifically conserved molecular patterns associated with pathogens, which are exclusively present on agents such as viruses, bacteria, parasites, and fungi. The main members of the pattern recognition receptor families are toll-like receptors, transmembrane proteins, C-type lectin receptors, cytoplasmic nucleotide oligomerization domain-like receptors, and gene-like receptors of the RNA helicase induction. Macrophages produce anti-inflammatory cytokines, such as IL-10 and TGF-beta, and inhibit inflammatory pathways mediated by toll-like receptors. Toll-like receptors initiate the signal cascade in innate and adaptive immune pathways. These highly phylogenetically conserved receptors represent a family of transmembrane proteins with two binding sites and an extracellular domain engaged in the recognition of potentially harmful antigens, both exogenous and endogenous, and an ectodomain that activates responses determined by antigen binding. Resveratrol regulates the expression of toll-like receptor 4. Therefore, resveratrol can interact with toll-like receptor-mediated inflammatory responses and chronic diseases related to toll-like receptor activation, including obesity, type 2 diabetes mellitus, fatty liver disease, Crohn's disease, rheumatoid arthritis, cardiovascular disorders, and neurodegenerative disorders. It inhibits TANK-binding kinase 1, a protein kinase serine/threonine involved in the antiviral response of innate immunity. Further studies have shown that resveratrol exerts an anti-inflammatory action by interacting with toll-like receptor 4, tumor necrosis factor receptor-associated factor 6, mitogen-activated protein kinase, and protein kinase B pathways in the populosaccharides-induced macrophages. Silent inflammation regulator 1 has a direct regulatory role in the functions of macrophages during inflammation, both in the secretion of cytokines and in the expression of cell adhesion molecules as intracellular cell adhesion molecules 1. Resveratrol also plays a key role in protecting the endothelium from inflammation, reducing the production of large amounts of colony-stimulating factor a pro-inflammatory cytokine essential for the differentiation and activation of pro-inflammatory macrophages, and which represents a specific marker of the formation of atheromatous plaques. Therefore, resveratrol can modify cell morphology, gene expression, ligand-receptor interactions, signaling pathways, and foam cell formation involved in atherosclerosis. In addition, resveratrol modulates the immune system response by affecting cellular levels of prostaglandin E2, which plays an important role in regulating the immune response. Finally, resveratrol upregulates the secretion of chemokines, such as cyclooxygenase 2, in various inflammatory diseases. By activating and stimulating macrophages and the production of IL-1 and IL-6, resveratrol also facilitates the systemic response to lesions and cooperates with tissue regeneration. 2. Natural killer cells. Natural killer cells represent about 15% of all circulating lymphocytes and play a critical role in the defense against pathogens and cancer. Natural killer cells express various pattern recognition receptors such as toll-like receptors, NLR, and RLRs. Activated natural killer cells release IFM-gamma, GM-CSF, TNF-alpha, or cytotoxic granules directed towards a target cell. Natural killers kill target cells through different mechanisms, inducing cell lysis, releasing cytoplasmic granules containing perforin, or by the secretion of several effector cytokines, such as IFN-gamma, IL-5, IL-10, IL-13, and GM-CSF, which trigger apoptosis in the target cell. Natural killer cells also secrete a variety of chemokines including the CC-chemokine ligand, such as CCL2, CCL4, CCL5, monocyte chemoattractive protein, macrophage inflammatory protein, chemokine oxychemokine ligand, and ILA. Natural killers, by interacting with other immune cells such as dendritic cells in areas of inflammation, modulate the innate and adaptive immune response and promote the T-cell response against tumors. Resveratrol exerts simultaneous effects on natural killer and T cells. It helps to increase the cytotoxicity of natural killer cells in vitro and in vivo, suggesting that resveratrol could be used as an adjuvant for cancer immunotherapy. An increase in the tumor lysis activity of natural killer cells was observed in a study evaluating the anti-infective properties of resveratrol in a mouse model of acute pneumonia. The resveratrol group showed increased alveolar macrophage infiltration, increased natural killer cell activity infiltration, reduced lung bacterial load, and decreased mortality. Spleen natural killer cells isolated from rats pretreated with resveratrol were more effective in inducing cell death against YAC1 culture target tumor cells. 3. Dendritic cells. Dendritic cells are a heterogeneous family of antigen-presenting immune cells. These cells are primary sentinels and share many characteristics with macrophages, which may also present antigen on major histocompatibility complex class E2 molecules, although not as effectively as dendritic cells. Resveratrol exerts potent immunomodulatory effects on dendritic cells' function. Resveratrol inhibits the expression of the co-stimulating molecules CD80 and CD86 and MHC class E2 molecules, leading to the suppression of phenotypic dendritic cell maturation, reducing the dendritic cell capacity to stimulate the proliferation of naive allogeneic CD4 plus T cells. Interestingly, due to the many molecular targets of resveratrol, many of which are involved in optimal dendritic cell maturation, resveratrol appears to exert even more potent immunosuppressive effects during the dendritic cell differentiation process. Dendritic cells from human peripheral monocytes incubated at various concentrations of resveratrol led to the creation of alternatively differentiated dendritic cells, unable to respond optimally to maturation stimuli, even in the absence of resveratrol. In addition, to inhibit surface expression of the co-stimulating molecules, these dendritic cells showed increased surface levels of transcripts similar to immunoglobulin-like transcript -3 and ILT4, two molecules closely associated with the tolerogenic dendritic cell function. Moreover, these dendritic cells can secrete greater amounts of IL-10, 2, 4, T and B lymphocytes. T and B lymphocytes cooperate with the APC in the immune response. However, once adaptive immune responses are triggered, Th1 and Th17 migrate from lymphoid tissue to the circulation, infiltrating infected sites and producing their cytokines, strengthening the activity of macrophages and neutrophils. Both innate and adaptive immunity can control inflammation and develop self and non-self discrimination. By T-cell receptor, T-cells are also involved in cellular recognition processes, along with proteins of the MHC, inducing possible apoptosis in a negative selection. To maintain immune balance, Tregs play a role in preventing the onset of autoimmune diseases and modulating the inflammatory response. Uncontrolled and abnormal activation of T cells is implicated in many autoimmune diseases, such as insulin-dependent diabetes, rheumatoid arthritis, systemic lupus erythematosus, and multiple sclerosis. Resveratrol may inhibit the activation of T cells and reduce the production of cytokines, preventing the autoimmune progression of the disease. Experimental studies have shown that mice with arthritis treated with resveratrol showed a significant reduction in the disease. Histomorphological evaluations have shown that the immune cells infiltrated into the joints were significantly reduced in mice treated with resveratrol compared to control mice, concluding that resveratrol may prevent the development of arthritis. Th17 cells are the main initiators of pro-inflammatory responses, recruiting neutrophils and macrophages at the site of infection, and through the production of IL-17 play an important role in defense against extracellular pathogens. Th17 also secretes IL-23, which controls the survival and maintenance of the Th17 phenotype and is responsible for the crosstalk between innate and adaptive immunity. Th17 cells produce IL-22 which, similarly to IL-17, is involved in many infectious and inflammatory disorders. In a clinical trial, male type 2 diabetes mellitus patients with hypertension were randomized to receive 350 mg/day for the first 6 months and 700 mg/day for the next 6 months of conventional grape extract without resveratrol, grape extract with resveratrol, or placebo of maltodextrin. At the end of the study, IL-6 was significantly decreased only in the group administered grape extract with resveratrol. In the placebo group, adabentin and IL-10 decreased significantly, while the IL-6/IL-10 ratio increased. Levels of several pro-inflammatory mediators were significantly decreased in the group taking grape extract with resveratrol compared to placebo. In addition, the administration of resveratrol suppresses the CD4+, CD25+ cell population among cells CD4+, reduces TGF-beta secretion, and enhances IFN gamma expression in CD8+ T cells both ex vivo and in vivo, resulting in immunostimulation. Resveratrol can inhibit T cells activation in autoimmune diseases and reduce Tregs suppressive function to inhibit tumor growth. Resveratrol induces the mitogenic proliferation of B cells at lower concentrations but inhibits it at concentrations higher than 10 microRm. Resveratrol does not appear to affect IgG or IgM production, but does induce a dose-dependent increase in the BCL2 expression, proteins that govern the permeability of the outer mitochondrial membrane, and can be either pro-apoptotic or anti-apoptotic. All effects of resveratrol on immune cells are summarized in Figure 2. 3. Resveratrol and COVID-19. In recent years, several viral pathologies have followed one another, such as severe acute respiratory syndrome in 2003, H1N1 in 2009, and Middle East Respiratory Syndrome in 2012. Since November 2019, a new type of coronavirus has caused a severe form of pneumonia, spreading rapidly worldwide since it was first detected in China, so much so that on 11 February 2020, the WHO declared the pandemic state. The disease induced by SARS-CoV-2 has been called coronavirus disease 19. This infection causes several symptoms from a dry cough to fever, from dyspnea to bilateral pneumonia. Patients with severe symptomatology can suffer acute respiratory distress syndrome, leading to admission to intensive care and, in several cases, even death. Many studies indicate that natural compounds have biological activities that counteract the onset of many viral infections, enforcing the immune response, limiting inflammation, counteracting the action of free radicals, and playing a potential virucidal role. Scientific evidence shows that resveratrol can exert virucidal action against several viruses. Several inflammatory mediators, such as NF-κB, TNF-alpha, and MMP9, were observed to be higher in chronic obstructive pulmonary disease patients than in healthy controls and were reduced after treatment with resveratrol. The severity of COVID-19 is a consequence of lung inflammation and injury caused by the viral infection, but also of an excessive inflammatory reaction related to the inefficiency of immune response triggered to control the virus, the cytokine storm. Resveratrol can interfere with viral replication through the inhibition of viral gene expression, viral nucleic acid, protein synthesis, and downregulation of various cellular transcription and signaling pathways. Resveratrol can also reduce the cytokine storm, acting on immune cells and limiting the lung-associated parenchyma injury. These properties have been demonstrated in infections induced by the influenza virus, respiratory syncytial virus, human rhinovirus, and MERS-CoV. Virocell studies have shown that resveratrol significantly inhibits the dose-dependent replication of SARS-CoV-2 with an effective concentration 50 of 48 mm. Adding 50 mm of resveratrol to cells after the virus absorption phase inhibited SARS-CoV-2 replication by 98%, suggesting that the molecule may also exert an effect on viral entry into cells. In Vero E6 cells, plaque-assessed viral replication was reduced by 50% and 90% respectively at resveratrol concentrations of 66 and 119 mm. Significant antiviral activity was observed up to 48 post-infection, a time point corresponding to approximately 5 cycles of viral replication, demonstrating the long-lasting antiviral effect of the compound. Resveratrol was then evaluated for antiviral activity in human primary bronchial epithelial cells obtained from healthy individuals. In addition to exhibiting direct antiviral activity, resveratrol showed inhibitory functions on the pathogenetic mechanism involved in the severity of COVID-19. These include the activation of unregulated NLRP3 inflammasome, dysfunction of the renin-angiotensin system, and stimulation of the quinine-kallikrein system. These inhibitory functions are mediated by the induction of the silent information regulator 1 protein that overregulates the expression of the angiotensin-converting enzyme 2 protein. NLRP3 inflammasome is a multiprotein complex that exists as a latent monomer in quiescent cells. Physiological activation of NLRP3 by stressors or pathogenic microorganisms facilitates the release of pro-inflammatory cytokines and promotes the induction of an adaptive immune response against bacteria and viruses. However, its uncontrolled dysregulated activation is involved in the pathogenesis of several inflammatory disorders. SARS-CoV-2 can activate NLRP3 directly through a viral protein viriporin 3A and indirectly through overproduction of IL-1 beta, a known causal factor for more serious complications of COVID-19. Autophagy can control activation of NLRP3 inflammasome through interactions with a variety of immune signaling pathways and the removal of endogenous inflammatory agonists. Resveratrol can activate silent information regulator 1, a decetylase that exerts protective effects on a variety of cellular functions, including apoptosis. In an experimental lung study of mice infected with a respiratory syncytial virus, silent information regulator 1 promoted autophagy-mediated processes leading to the activation of dendritic cells, the presentation of viral antigen to T cells, and an effective antiviral immune response. Thus, through downregulation of the excessive inflammatory response demonstrated in other viral infections and upregulation of silent information regulator 1 activity, resveratrol can inhibit NLRP3 activation, leading to autophagy. A study by Terrell et al. Confirmed that resveratrol has a potent antiviral effect against COVID-19. This phenolic compound greatly reduced the replication of SARS-CoV-2 not only in Vero E6 cells, but also in a human bronchial epithelial cell model. Moreover, it actively interferes with the infectious replication cycle of the virus and exerts antiviral activity up to about 5 replication cycles of SARS-CoV-2 in vitro. The study hypothesizes that resveratrol interferes with the early stages of virus replication, thereby reducing the chance of productively infecting a cell. In addition to the antiviral properties targeting SARS-CoV-2 replication, resveratrol may also be suitable for moderating the exacerbated inflammatory response seen in patients with COVID-19. Resveratrol, as an antioxidant, is found to prevent the formation of reactive oxygen species, prevent epithelial airway remodeling by upregulation of silent inflammation regulator 1 and superoxide activate dismutase. Due to the low bioavailability of resveratrol after oral administration, an effective antiviral dose against respiratory viruses in humans is unlikely to be achieved by this route of administration. Therefore, alternative modes and routes of administration have been investigated. Promising strategies for antiviral treatment are the use of resveratrol aerosol suspension sprays, dried microparticles co-spray, or nanotechnological approaches. While improving bioavailability, some of these techniques allow direct and local pharmacological administration of resveratrol to the primary active site of replication of SARS-CoV-2 by inhalation-based systems currently considered a favorable future strategy for the treatment of COVID-19. Therefore, resveratrol might not only be a promising candidate for inhibiting viral replication at the onset of infection, it may also alleviate the symptoms of the disease later in infection. 4. Resveratrol and metabolic syndromes. Obesity represents an authentic epidemic. The prevalence of overweight and obese people has increased since the 1970s, and in 2016 about 39% of the world's adult population was overweight. Obesity has serious health effects, increasing the risk of developing type 2 diabetes, cardiovascular disease, dyslipidemia, Alzheimer's disease, and even certain cancers. Reducing obesity requires major lifestyle and dietary changes to avoid surgery or medication. Resveratrol showed several positive effects on the management of obesity, inducing mitochondrial biogenesis and oxidative phosphorylation via the activation of peroxisome proliferator activated receptor gamma coactivator alpha via silent inflammation regulator 1 decidylase, leading to improved insulin sensitivity, and by increasing the expression of silent inflammation regulator 1, a reduction in the size of adipocytes, showing an antilipolytic effect. A recent study on a mouse model fed with a high-fat diet showed that resveratrol supplementation can improve the composition of the intestinal microbiota and enrich the pathways involved in generations of small metabolites, despite the low bioavailability of this polyphenol, contributing significantly to the prevention of metabolic syndrome. Diabetes mellitus, a group of chronic metabolic diseases characterized by hypoglycemia, is one of the most serious consequences of obesity, caused mainly by a total or partial dysfunction of the Langerhans islands of the pancreas, responsible for the production of insulin, the hormone that regulates blood glucose levels. In 2017, the disease affected about 451 million people, and this number could increase to 693 million by 2045. Often, the various vascular complications caused by hypoglycemia, together with oxidative stress and inflammation, are the primary reason for morbidity and mortality associated with diabetes. Resveratrol, however, has potent antioxidant and anti-inflammatory activities, acting directly on lipid peroxidation and removing superoxide free radicals. By activating silent inflammation regulator 1, resveratrol induces the decetylation of the transcription factor 4-kidbox-O, involved in cell apoptosis, and increases the expression of superoxide activate dismutase, which has a key antioxidant role. The absorption of beta cell antigens by islet-resident dendritic cells, presentation to naive T cells, and promotion of Th1 development will activate B lymphocytes, which will create autoantibodies against beta cells. Th1 will also trigger macrophage and neutrophil migration to the islet, which will increase reactive oxygen species and enhance beta cell death. Resveratrol works through a binding to C-chemokine receptor 6, which inhibits Th1 cell migration, creating a complex with insulin, which stimulates glucose intake. Resveratrol also reduces reactive oxygen species and boosts antioxidant capacity by inhibiting apoptotic cell damage during oxidative stress via silent inflammation regulator 1. Resveratrol also aids in the regeneration of beta cells in the pancreas. A clinical study on 13 patients with type 1 diabetes was performed by Movid et al. In 2019, these subjects were given resveratrol in 500 mg capsules twice daily for 60 days. Fasting blood glucose and hemoglobin A1c levels were significantly reduced with the supplementation of resveratrol, also leading to a decrease in malondialdehyde, a marker for oxidative stress. This confirms the strong antidiabetic and antioxidant effects of resveratrol, which can be considered a valid aid for the prevention and management of metabolic syndromes. Moreover, a meta-analysis of 11 individual studies showed that resveratrol consumption significantly reduced fasting glucose, insulin, glycated hemoglobin, and insulin resistance levels in a subgroup of diabetes patients. However, further clinical and preclinical evidence, with larger samples, together with further studies to improve its bioavailability, are needed to confirm the efficacy and safety of this polyphenol in such delicate pathologies. Resveratrol and neuroinflammation. Neuroinflammation is the brain response to an insult, infection, or disease which aims to remove or inactivate potentially damaging agents to brain tissue. This response is mediated by the interaction between two cellular systems of the central nervous system, whose activation leads to the triggering of certain immune cells of the hematopoietic system. Neuroinflammation is one of the major contributing factors in exacerbating neurodegeneration and depression, which, especially in combination with chronic stress, can increase the level of pro-inflammatory cytokines such as TNF-alpha and IL-1 beta, leading to the development of diseases such as Alzheimer's disease and Parkinson's disease. Many studies have shown the neuroprotective and immunomodulatory role that resveratrol plays against neurodegenerative diseases. Resveratrol is one of the major activators of silent information regulator 1, a nuclear protein that deacetylates transcription factors that govern metabolic central pathways, whose key role in the management of AD has been widely evaluated. When it is overexpressed in the brain, it reduces this neurodegenerative disease, as it directs the division of amyloid precursor protein from beta-amyloid production through the activation of alpha secretase, and deacetylates the tau protein, thereby reducing the formation of neurofibrillary tangles. In a 2016 study in which a memory and learning deficit was induced in a mouse model by microinfusion into the hippocampus of beta-amyloid 1-42, the main constituent of amyloid plaques, had formed during the exerts of AD, higher dose of resveratrol was shown to significantly improve the memory of rats and to decrease the expression of apoptotic and inflammatory cytokines, such as IL-1 beta and IL-6, acting on T cells. The inhibitory effect of resveratrol counteracts the activity of phosphodiesterase 4, which controls intracellular levels of cAMP. In a rat model with a spared nerve injury, performed to simulate symptoms of neuropathic pain, resveratrol has shown neuroprotective effects, inhibiting the inflammatory response of neuropathic pain, as well as relieving mechanical allodynia, one of the main symptoms of this chronic disease, through an administration of 300 microg/day, improving autophagy and downregulating the expression of triggering receptors expressed on myeloid cells 2 in microglial cells, identified as one of the critical factors in inflammation in the nervous system. A clinical study in patients with AD showed that after 52 weeks of resveratrol administration, the levels of MMP9, a matrixin involved in the degradation of the extracellular matrix, in the cerebrospinal fluid of these patients were significantly decreased. MMP9 appears to be implicated in several brain pathologies, such as neurodegeneration and neuroinflammation, because it regulates the permeability of the blood-brain barrier through the release of cytokines and free radicals. The reduction in this enzyme shows that resveratrol administration reduces brain permeability and, as a result, the infiltration of leukocytes and other inflammatory agents into the brain, as well as inducing an adaptive immune response that may increase brain resilience to amyloid formation. 6. Resveratrol and infertility. Infertility is a widespread condition in industrialized countries where it affects 15–20% of couples. It is defined as the inability to reach a spontaneous pregnancy after at least 1 year of regular and unprotected sexual intercourse. Male infertility may include abnormal sperm parameters or a combination of all three, or azoospermia. Male infertility causes can be divided into 4 categories, with idiopathic infertility accounting for up to 25% of cases. Most studies on infertility treatment, both in vitro and in vivo, have focused on the mechanisms of oxidative stress that could cause a lipid peroxidation, with altered membrane fluidity and permeability, resulting in decreased sperm motility and reduced sperm ability to interact with the egg cell, protein modification resulting in reduced adenosine triphosphate production and increased fragmentation of sperm, DNA. Male infertility may be associated with testicular immune microenvironment disorders. The testicular immune microenvironment consists of common immune cells associated with other cells of the organ, all involved in testicular immunity. These cells include macrophages, T cells, dendritic cells, and mast cells associated with Leydig cells and Sertoli cells. Several studies have shown an overproduction of pro-inflammatory cytokines dependent on alterations of this microenvironment, as in the case of varicocele. The anti-inflammatory action of resveratrol could inhibit this cytokine hypersecretion, counteracting infertility. A clinical study evaluated the protective effect of resveratrol in counteracting infertility damage induced by benzopyrin, a highly toxic and carcinogenic polycyclic aromatic hydrocarbon originating from cigarette smoking, smoke from some factories, and even from burned food. The study was conducted on 30 healthy men with an average age of around 32 years at the Division of Andrology and Endocrinology of the University of Catania. Sperm of 15 men were incubated with increasing concentrations of BARP at 37C. The effects of BARP on sperm motility and biofunctional parameters were assessed after the incubation period. Once BARP response concentration was established, the sperm of the remaining 15 men were exposed to only 15 mcm/ml BARP and / or 15 mcm/ml resveratrol. BARP resulted in a progressive decrease in sperm motility and abnormal compact chromatin. Resveratrol showed its antioxidant and anti-inflammatory effect by decreasing the percentage of sperm with altered chromatin compactness. It also improved the total and progressive motility of sperm and lowered the oxidative stress indices. Some studies have shown that resveratrol appears to be able to enhance the estrogenic effects of hormones, and therefore is a modulator of female reproductive function. Estrogens are also secreted by human Leydig cells in the testicles where they play a paracrine regulatory function. Resveratrol is a phytoestrogen in females, with a chemical structure comparable to that of estrogens. Resveratrol shows a cytoprotective effect on oocytes with antioxidant and antipoptotic effects. In males, resveratrol affects reproductive function in 3 ways: by improving testosterone production, by triggering penile erection, and by improving spermatogenesis. This suggests a possible role of resveratrol in male fertility. This polyphenol activates antioxidant enzymes such as catalase and superoxide activate dismutase, involved in the lipid damage of human sperm during the cryopreservation process. In addition, resveratrol shows an anti-inflammatory effect by decreasing the activity of inflammatory molecules, such as cyclooxygenase 2, inducible nitric oxide synthesis, a linking enzyme between immune and endocrine cells, and B-cell NFKB. Regulating anti- and pro-apoptotic mediators also protects cells from DNA damage and apoptosis. Spermatogenesis, therefore, can be extremely sensitive to compounds that interfere with mitochondrial energy metabolism and respiratory control. Any alteration in the regulation of metabolic behavior of these cells may impair the normal development of spermatogenesis and, consequently, male fertility. It has been proposed that mitochondria also play a role in the sperm degenerative process, thus ensuring that good quality meiotic products enter the spermatogenesis process to produce quality, mature sperm. Interestingly, resveratrol has been shown to increase the mitochondrial number and activity in several cell types, such as muscle cells and granular cells. Resveratrol enhances mitochondrial function by activating silent information, regulator 1. Thus, it can regulate cell biology, metabolism, and fate at various levels. 7. Conclusions. The broad spectrum of action of resveratrol makes this phenolic compound able to counteract the onset of different diseases or to contain their symptoms, thanks to the interaction that resveratrol has with immune cells and its action on silent information regulator 1. This polyphenol can modulate different inflammatory and non-inflammatory responses, inducing protective effects on organs and tissues. The anti-inflammatory, antioxidant, antipoptotic, neuroprotective, cardioprotective, anti-tumor, and antiviral power make this compound a possible adjuvant of classic molecules 2022, 27, 424 13 of 19 pharmacological therapies in public health management. New experimental and clinical studies are needed to improve the bioavailability of this nutraceutical and to evaluate the most appropriate route of administration to maximize its efficacy. In addition, broadening the sample size in the various clinical trials could provide a clearer and more complete picture of the efficacy and safety of resveratrol.